Okay. Unit 1, Archaebacteria. We will quickly see the summary what we have learned during our class, previous class. In this, we have two sections. First, the general properties of Archaebacteria, and then specific group of Archaebacteria genus, group of Archaebacteria, and their features. Archaea itself, it indicates that it is an ancient group of organism which has vast varieties, difference in their morphology and physiological conditions. They are having different shapes and reactions. That means a certain member of this group is a gram positive and other are gram negative. Shapes are also spiral, rod, and pleomorphic. Many different shape are found in archaebacterial group of organism. It has been believed that it is the first form of life on Earth. The first fossil of this group was found before 3.5 to 3.8 billion years ago in a form of stromatolites or sedimental rocks, which are incorporated with minerals with microbial mats. You can see that here archaebacteria are divided or grouped separately from the prokaryotes as well as eukaryotes. On a study in 1977, Carl Bulls and his colleagues studied RNA sequence of this group, and he distinguished them, this group, then prokaryotes and from eukaryotes, as there are a difference in their 60S rRNA sequence, which is also known as signature sequence. There is one hypothesis regarding this, that is endosymbiotic hypothesis. The first evidence of eukaryotic cells. The ancestor of eukaryotic cells may have developed some fusion of ancient bacteria and archaea, possibly a gram-negative bacteria host cells that has lost its cell wall engulfed by and archaeons to form an endobiotic association. Means what? It is believed that whatever the eukaryotes and their organelles are there, these organelles may be as a result of endosymbiotic hypothesis of endosymbiotic relation of certain prokaryotes or archaebacteria with eukaryotic cells. For this, there is very well-known theories or hypothesis like chloroplast is a fusion of some photosynthetic bacteria inside the host cell, and due to certain mutations and after a long time, certain properties has been mutant or lost by that photosynthetic bacteria, and just it is able to go for photosynthesis, and now it is now known as chloroplast. The same way mitochondria, even Golgi complex, they all are so origin of archaeons. This is the comparison between the bacteria, archaea, and eukarya. Archaebacteria have certain characteristics which resembles to bacterial cells, and some of them are quite similar to eukarya. In this, you can see that the cell wall, almost bacteria have peptidoglycan containing muramic acid, while in archaea they have variety of types of cell wall compositions and no muramic acid, same like eukarya. In this, when we see about membrane lipid, normally in bacteria, ester-linked straight chain of fatty acids you will find, while in archaea, ether-linked branch aliphatic chain will be present, and in eukarya, it is still linked to straight chain fatty acids. So in this, when we talk about membrane cell membrane lipid content, it is quite different in compared to bacteria as well as eukarya. One by one, when we see this, all the characteristics, you will find that certain characters are very similar to bacteria, while other are similar to eukarya. And at the as a end of this comparison, we can say that archaea are not a bacteria or not an eukarya. It is quite third kingdom, which is different from both of them. Normally, some of these archaebacterial cells are single and certain prepare a filamentous or aggregated structures. Their range of diameter is also quite different from 0.1 micrometer to 15 micrometer. So, some filamentous can grow up to 200 micrometer length also. Their multiplication or reproduction is normally in a form of binary fission, budding, or fragmentation. Same way, archaea also diverse physiological properties, like they are aerobic, facultative anaerobic, strictly anaerobic. They have wide range of nutrient uptake also. They are chemolithotrophic to organotrophic. Means you can see that how much variations are there in their mode of nutrition. Same way, archaebacteria's certain genus are mesophilic, and while other are hyperthermophilic. That means they can grow above 100 degree. Their habitats are extremely different. Some are extreme, like the north and south pole. They are certain, you will find it high temperature environment. It is also present in aquatic or terrestrial habitats. They some are strictly observed in anaerobic condition or hyposaline environment. Recently, archaea has been also discovered in cold environment. So you can see that you can guess that extreme environments where we should not think about, or our minds stop thinking about that there may be life. At that place, you will find a certain group of organism which belongs to archaebacteria. Even a few are present symbiotically in animal digestive system, as animals, especially ruminatic animals, have a sector type of stomach, and in that methanogens are frequently present in symbiotic way. Uh, there are distinguishing characteristics of archaebacteria. Their cell wall, lipid membrane, other informational process, means whatever the biochemical characteristics are there, biochemical machineries are there, and their physiological adaptation to an extreme environment is also quite different. Cell wall. One by one, we will see each and every part. Cell wall, although archaebacteria can stain gram positive or gram negative, depending on their cell wall thickness and cell composition, the structure and chemical composition is quite differ from the bacteria. Their construction of archaeal wall is also in a variations. Many gram positive archaea have a wall with a single thick homogeneous layer, which resembles that in gram positive bacteria. Thus, they are gram positive archaea. While some are gram negative archaea, lacks the outer membrane and complex peptidoglycan layer of gram negative bacteria. Instead of a usual surface layer of protein or glycoprotein subunits, they have some different cell composition. Gram positive archaea have a variety of complex polymers in their wall. For example, methanobacterium and some other methanogens have walls containing pseudomurine, a peptidoglycan like polymers with L-amino acid, and some have N-acetyl telisomuramic acid instead of N-acetyl muramic acid and 1,4-big 3 glycosidic bonds instead of 1,4 glycosidic bonds. While in methanoscarin and halococcus, lakes pseudomurine and contain a complex polysaccharides in their cell wall. While in gram-negative archaebacteria have a layer of protein, glycoprotein outer membrane is lacking. The layer may be thick 20 to 40 nm. Sometimes there is a two-layer, a sheath surrounding an electrodense layer. The chemical content of these walls varies considerably. Some methanogens like methanolobus and halobacterium and several extreme thermophiles have glycoprotein in their walls. While in contrast, other methanogens, desulfurococcus and extreme other organism contain protein walls. This is the structural information about pseudomurine. You can see that it contains an acetyl talcomuramic acid and an acetyl glycosamine instead of muramic acid, substitute of an acetyl muramic acid of the peptidoglycan. This is the diametric difference, diagrammatic difference of gram-positive and gram-negative archaebacteria. In a summary, gram-negative, no outer membrane, no cell wall, thick protein or glycoprotein coat. While gram-positive cell wall is made up of pseudomurine and other complex carbohydrates. Okay. Now we will see archaebacterial cell membrane.